As we just laid out, there is directionality in protein. Here is our building block, our individual amino acid, and then if we put a whole bunch of amino acids next to each other and connect them along their backbone, this being their backbone right here, then we're going to see the amino group to the central carbon to the carboxyl group of one amino acid being connected to the next amino group to central carbon to carboxyl group, and so on and so forth along the way. If we look at a couple of examples here, we have two amino acids that have already formed a bond in the backbone and one that is being added to the chain. So you can imagine right now we are inside the ribosome. These two amino acids have already been added to the polypeptide, and this one is being called in via whatever codon matches up with this amino acid in the codon chart. Okay, so these two are already connected via what we call a peptide bond. And every single time that we connect one amino acid to another during translation, we're creating a peptide bond. And the way that we create a peptide bond is by linking the carboxyl group of one amino acid to the amino group of the neighboring amino acid. And the way that we do this is we take the hydroxyl group off of the carboxyl group and we take the hydrogen off of the nitrogen. And those two things come together to form water, which falls out. We call this a condensation reaction because water is formed, condensation, water. And now we have a bond ready to be formed with this carbon, a bond ready to be formed with this nitrogen, and those two come together to form a new peptide bond. So every single time you add a amino acid to the polypeptide chain, every single time you add a building block to the protein, you're creating one molecule of water in the process, or releasing one molecule of water in the process. And this entire process of translation is all happening in the backbone. The backbone of one amino acid is bonding to its neighbor. We haven't yet talked about the R groups or the side chains. We'll do that in a later video. My example here just has 3 amino acids. Let's look at a slightly more detailed example. So again, here another way of representing the amino acids is if we ionize the carboxyl group and ionize the amino group here, but the exact same rule applies. We're still going to take H2O out here, so we're still going to take this oxygen, and we're going to take 2 hydrogens, and we're going to create a new peptide bond between this carbon and this nitrogen into a neighboring amino acids plus water that you see there. And all along the chain of amino acids, remember proteins are generally hundreds to thousands of amino acids long. So if we looked at the backbone of any amino acid, we would see this repeating sequence. There's one amino acid's backbone, bound to another amino acid's backbone, down to another amino acid's backbone, and another, and another, and another. And every time that we see them connected, we're seeing them connected via the peptide bond, which is indicated in red here all along the way. The only thing that differs between these amino acids is their R group. The backbone stays the same the entire time. And the directionality here is indicated at the start and at the end. We have the N terminus at the start of the protein and the C terminus at the end of the protein. And again, those names come from N for nitrogen in the amino group of amino acid number 1, and C for carbon in the carboxyl group of the last amino acid in the chain. This is the detailed version of how we would show a polypeptide with every single atom drawn out. Oftentimes we represent a polypeptide like this instead. Oftentimes we would say, okay, here's the N terminus, here's where it starts, here's where it ends, and this is each amino acid in the chain. So we just use the abbreviation or the full name of the amino acid instead of drawing out every single atom like you see up here. And if you look back at your worksheet of the amino acid proteins, you can then use that information to go ahead and get an idea of what these individual puzzle pieces look like. So for example, let's do glycine. So glycine is abbreviated GLY. And if you look on your worksheet, it's the very first one up there on the top left. And so glycine is an amino acid with the backbone that looks like all of our amino acids. Central carbon bound to an amino group and a carboxyl group, and its R group happens to just be a single hydrogen. So if you were looking at a polypeptide chain represented like this and you wanted to know what glycine looked like, you would just look on your worksheet to look at that amino acid and see, okay, there it is. It looks like this. It's got an R group like that. Versus, what's another one here? Alanine. Alanine, the next one, ALA. If you look on your worksheet, that's right next to glycine, so it's one along. And again, the backbone is exactly the same as every other amino acid, but the thing that differs there is that R group. The thing that differs is that bunch of atoms that comes off of the central carbon facing up in this example. And so that is right here. a CH3 or a methyl group instead of a single hydrogen coming off. Or next one along, serine. Serine, can you find it on your worksheet? That one's in the middle of the page, all the way over on the left. Yep, so backbone identical, but our group differing there. Looks like this, and so on and so forth. So whichever way you are presented these molecules, you can work one way or the other to figure out what the molecule looks like. And once you know what the molecule looks like, you can start to think about the properties of this molecule and how it will interact with other molecules, intermolecular interactions. And so, for example, something like right here, you've got a non-polar covalent bond, and that's going to behave differently than something like this, which is a polar covalent bond and involves an ionized atom here, or here, polar covalent bond. And we'll talk a little bit more about that a little bit later.